A big hello to all my future doctors out there. I hope you're doing great, my dear children, and are super ready to wear your shining armor, that is the stethoscope and the doctor's coat and everything. Well, and I also hope that you have watched the previous video of day 1 where I dealt with two important chapters, that is molecular basis of inheritance and also principles of inheritance and variation. If you have not watched it, please go and watch it. And today we are going to cover these 3 chapters, that is biotechnology principles and processes, biotechnology and its application, and also human health and diseases. So if we ask which chapter gives you the most amount of marks, it is the first one, that is biotechnology principles and processes, my dear children. So you have to be very sure that you are practicing enough amount of questions. Previous year I think 5 questions came up, or basically the tentative thing that we expect is 5 questions would be coming in this year as well. So let's hope that, and let's give our best for that. And as I always say, whenever I show you a question, pause the video, try to do it by yourself and then check your answers with me. And if you're in, my dear children, do let me know that you're in. Yes ma'am, I will be taking up this challenge, and today I have completed day 1. Now I'm going for day 2. So do let me know whether you have completed day 1 or not. Now, biotechnology principles and processes. The first question that we have from this chapter is this: which one of the following is a case of wrong matching? Somatic hybridization, fusion of 2 diverse cells. Is it somatic hybridization? Yes, my dear children. What happens in somatic hybridization is that you remove the cell of a plant, right? It has a cell wall. So basically you remove the cell wall. Whatever is left behind, that is your cell membrane plus the cytoplasm and everything together, you call this as protoplasm. So you fuse the protoplasm of 2 diverse cells, and this is known as somatic hybridization, okay? And you get somatic hybrids out of it. Tomato and potato was mixed like that, yeah? So yes, this is somatic hybridization. That is totally correct. You have to find what is a wrong matching. Now, vector DNA is site for tRNA synthesis. I suppose vector DNA is something that would be basically carrying my gene of interest. For example, there's a DNA, I am putting my gene of interest there. This particular vector DNA is going to take this gene of interest to the place where I actually want to put, for example, a host organism. So vector DNA is that something that would be carrying my gene of interest. It's not a site for tRNA synthesis. So this one is a wrong matching, but we have to go ahead with next option, right? We have to see. Now, micropropagation, in vitro production of plants in large numbers. Yes, that is known as micropropagation, yeah. And callus is the unorganized mass of cells produced in tissue culture. So in tissue culture, what do? We take a part of a plant which are having cells that divide way too much, okay? They contain high amount of meristematic cells, yeah? And then what do? We put it inside a solution that contains a good amount of nutrients, so that the cells can easily divide and form something that is known as callus. Now this callus, I'm going to put it in rooting hormone and shooting hormone, so that small root and small shoot can come in and it can form a whole new plant. So I hope you got that, okay? The part that we take from the plant is known as X plant. So it is not the part of a plant now, it is X, okay? So it is known as X plant. Remember that as well. So this and this is correct. The only wrong option that you have is vector DNA. It's not a size for the RNA synthesis, my dear children. Next question: choose the correct pair from the following. Chalo. Ready? Do we have the Josh? Let me have the Josh. Okay. Ligases joins the 2 DNA molecules. Is it? Yes, they ligate the 2 DNA molecules. So for example, in genetic engineering, you cut the DNA, right? So this cutted DNA which is there, it cannot stay like that. You have to stick something. So basically, ligases are going to act like the super glue out there, and that is absolutely correct, that they join the 2 DNA molecules. Now polymerases breaks the DNA into fragments. That's something very odd. According to what I have learned, ma'am, DNA polymerase, RNA polymerase, everyone is responsible for making up something, not breaking up, okay? So polymerases are not for breaking. This would be a very wrong statement. Nucleases separate the 2 strands of DNA. Again, nucleases, my dear children, are something that would be cutting the nucleic acid, for example, DNA or RNA. It's not something that would be separating the 2 strands of DNA. That is usually done by helicases, okay? That is usually done by helicases. You're talking about nucleases. Nucleases, as in it is going to cut nucleic acid. And what is nucleic acid? Deoxyribonucleic acid. deoxyribonucleic acid 1 and then ribonucleic acid 2, that is DNA and RNA, my dear children. Okay, so it's not about separation of two strands, this would be wrong. Exonucleases makes cuts at specific position within the DNA. You know, we had two type of nucleases, okay, that is exonucleases and endonucleases. Exonucleases as in you're talking about the last portion, the ending portion. You're going to cut DNA from the ends, okay. Endonucleases means they are going to cut DNA from within. So here it is written exonucleases makes cuts at specific locations within the DNA. That would be wrong because they are the one that makes cuts at the ends, towards the end. So this would be wrong statement. What is the only one that is correct? It is this. It is what? This. So your answer is A. Fine. Next question. The sequence that controls the copy number of the linked DNA in the vector is termed as. For example, dear children, I have a PBR 322 plasmid. Okay. Now over here, I am going to put my gene of interest. So this is going to be my gene of interest. Which is that something? What is that something in this particular PBR 322 plasmid? 322 plasmid that will be controlling the copy number, how many copies of this particular gene of interest could be made, or basically as a whole, how many copies of this particular plasmid would be made. Who controls it? It is the origin of replication, ORI site. ORI site is where the DNA polymerases will come and attach and do the replication. It is also the site which will decide how many copy numbers would be made. So sequence that controls the copy number of the linked DNA in the vector is termed as selectable marker? No. Palindromic sequence is something that you can read equal same from both the sides. For example, Malayalam. Any Malayalis out there? Malayalam. I am a Malayali. So if you read from here, it's Malayalam. If you read from here, that's also Malayalam, right? Now, recognition sites are those sites where the endonucleases, okay, restriction enzyme will recognize that site and cut it. They have specific sites only where they work. Okay, so these sites are known as recognition sites. So it's ORI site that is important. Origin of replication, my dear children, please remember it. It's origin of replication. Cool? Works? Okay. If it works for you, it works for me. Now, in gel electrophoresis, separated DNA fragments can be visualized with the help of. So, in gel electrophoresis, what are you doing? You are basically separating the DNA, right? But this DNA, is it visible to you? No. Baba, you have to stain it with something that is known as ethidium bromide. And ma'am, now it will be visible, right? No. There's a twist in the story. You have to see it under the UV light. When you see it under the UV light, it will appear to you as bright orange color. It will appear as bright orange color, my dear children. So, acetocarbon in bright blue light. Bright blue light. Bright blue light. No. Ethidium bromide in UV radiation. Yes, we got the answer. Okay, acetocarbon in UV radiation. No. Ethidium bromide in infrared radiation. No. Your answer is B. Got it? Now, next, given below are 4 statements pertaining to separation of DNA fragments using gel electrophoresis. Identify the incorrect statements. DNA is negatively charged molecule. Okay. Before I get ahead, I want to explain you how does gel electrophoresis work and you tell me the answer then. Okay. So, in gel electrophoresis chamber, first thing that you have to know is DNA is negatively charged. What? DNA is negatively charged. Remember, it has a phosphate group, negative charge, yeah. So, it has a negative charge. So, my guys, over here, the DNA is kept. Okay. Suppose, over here you have cathode. Okay, and over here you have anode. Now, which one is positive, which one is negative? Cations are positive. Cats have pos. So, cations are positive. So, but cathode is negative. But cathode is the opposite, it is negative. Anions are negative, but anode is positive, my dear children. So, what is happening? Basically, when I am going to put my DNA over here, which is negatively charged, will it stay here or will it move away? Obviously, the things that are same, it will repel, okay, it will repel. So, they basically move away from it. Now, who is the one that is going to move the fastest or the farthest? It will be the smaller fragments. Smaller fragments, they will move really fast and they will reach this particular side, yeah. And now, anything else? Basically, when I say they are moving, it's not like, first of all, in gel electrophoresis, you are using the gel, right, from the seaweed and things like that. Do you think the DNA is moving on the surface level? No. It is actually having a sweeping effect. That is, this gel that we used has a sweeping effect. The DNA which is there, it sieves through. Okay, just like sieving effect as in, for example, when you sieve your clothes and all, how it goes, right? Similarly over here, it is going to go from within, not from the surface. Always remember that. And now I think probably you can answer the question. So given below 4 statements pertaining to separation of DNA fragments using gel electrophoresis, identify the what? Incorrect or correct one? First of all, underline this. You have to find the incorrect one. Okay, so DNA is negatively charged, agreed, and so it is loaded on the gel towards the anode terminal. Do you think it is put on the anode terminal? Wrong. First statement itself is incorrect. Okay. Now DNA fragments travel along the surface of the gel whose concentration does not affect the movement of the DNA. Jai Baba, it cannot travel through the surface of the gel. Wrong. Now smaller the size of the DNA fragments, larger is the distance it travels through it. Yes, it is correct. Okay, now pure DNA can be visualized directly by exposing UV radiation. What about ethidium bromide? What is it doing? You need a stain to visualize it, right? So you need a stain. It's not directly, you cannot visualize it directly by exposing it to UV radiation. So this would be a wrong statement again. So I have got 1, 2 and 4th one as wrong. 1, 2 and 4th. This one would be your answer. Yes, yes. Next question. Commonly used vectors for human genome sequencing are? For human genome sequencing are, what vectors do you use for human genome sequencing? You have to use something big that can accommodate the huge, big human genome that is there, parts of big things. So is it tDNA? No. Is it expression vectors? No. It is TA cloning vectors? No. What is it? It is BAC and YAC. Straight. Okay, you don't need explanation to any options. Just remember BAC and YAC. Bacterial artificial chromosome, yeast artificial chromosome, my dear children. Why them? Why them? Because they can accommodate larger fragments. They can accommodate larger fragments, which is very much important in sequencing of human genome. So your answer becomes what? B. Works? Works. Works, ma'am. Okay, tell me one more thing. Hepatitis B vaccine. Okay, since I remember it, I am telling you right now. Hepatitis B vaccine, or basically hepatitis vaccine, was made with the help of what? With the help of what? Yeast, artificial chromosome, yeast. It was yeast. With the help of yeast only this particular vaccine was being made. Remember that. Okay, it was yeast artificial chromosome that was being used. Now, the colonies of recombinant bacteria appear white in contrast to blue colonies of non-recombinant bacteria. Because of what? The colonies of recombinant bacteria, recombinant as in the one that has a newer combination. I have made some changes in there. Okay, so for example, I have a plasmid. This is a plasmid, my darling children. Okay, and this plasmid had a gene, that is LAG-Z gene, and Z codes for beta galactosidase. So it is still coding for that. And now when I put X-gal, a substrate that could give color, okay, when I put X-gal, it reacts with it and gives blue color colonies. Blue color colonies. This is the usual scenario. Now what Jasmine ma'am going to do is really bad. She's going to take the plasmid and she's going to put the gene of her interest. Where? In the LAG-Z gene. Jasmine ma'am is so cruel. She put it over here. Okay. Now what will happen? Do you think it can code for beta galactosidase, my dear children? No. In this case, even if I put X-gal, will you see any change in the color? No, ma'am. There is no enzyme to bring out any changes. You get it? So basically, the recombinant, this is what? A recombinant. This has the new combination. This is what? A non-recombinant that we have. Okay, now tell me, the colonies of recombinant bacteria appear white in contrast to blue colonies of non-recombinant bacteria. Because of insertional inactivation of alpha galactosidase in the recombinant bacteria, do you think any alpha galactosidase is getting inactivated? No. Inactivation of glycosidase enzyme in the recombinant bacteria. Let's go ahead. Non-recombinant bacteria containing beta galactosidase. The colonies of recombinant bacteria appear white in contrast to blue colonies of non-recombinant bacteria because non-recombinant bacteria contains what? Beta galactosidase enzyme, my dear children. Yes, is that true? Yes, that is true. Over here is this glycosidase enzyme being mentioned. It is glycosidase? No, it is galactosidase. It is what? Galactosidase. So this is wrong. Okay, and here insertional activation of alpha galactosidase. Array alpha, there is no alpha, there is beta. This is beta galactosidase. So which is why your answer becomes C, my dear children. Be very careful. first, while even when I read it, okay, I was like glycosidase, this is the answer. Okay, but then I went ahead and I saw it is written, this made more sense to me. Then I read it back again, then I realized, oh, it is glycosidase, not galactosidase. You get it? So basically you have to make sure you're reading the options. By reading the options, sometimes you get to know the answer as well. Okay, you come in the safe zone. So please do that. Yeah. Now next thing, the given figure is the diagrammatic representation of E. coli vector BBR 322. Which one of the following options correctly identify its components? So over here, I'll just tell you, then you tell me. Okay, so it is the origin of replication, where the DNA polymerase will come and attach and replicate. This is the ROP site, that is it is a very important protein for a initiation of replication to happen, ROP. And in ROP site, my dear children, there is a recognition site for PVU2. There is a restriction enzyme, and this restriction enzyme can make a cut over here because it has a recognition site over there. It's PVU2. Okay, in the ROP site you have it. Now on both the sites you have two selectable markers. One is tetracycline resistant gene and one is ampicillin resistant gene. That means a PBR 322 having both of these genes, my dear children, they are resistant to action of ampicillin and also tetracycline. That means they can be easily grown in both of the medium because they are resistant to it. But there is a twist to the story, that is, in the tetracycline resistant gene, my dear children, you have recognition site for two enzymes, BAMH1 and SAL1. Suppose in the question it says that you are cutting with BAMH1. If you are cutting with BAMH1, basically you are going to put your gene of interest over here. Do you think then it will be tetracycline resistant anymore? It would not be tetracycline resistant anymore. Please make it understandable. Okay, please have it in your head. So two enzymes, BAMH1 and SAL1. Now going ahead, you have PVU1, here you had PVU2, right? So there's PVU1 and PST1, both of them have the recognition site over here. In between both of these, you have three enzymes recognition site, EcoR1, CLAV1 and basically HIND3. Okay, now you have to tell which one of the given options correctly identify its certain components. ORI, original restriction enzyme, it's what? Origin of replication. ROP, reduced osmotic pressure. HIND3 or basically EcoR1, selectable markers. No, they are not selectable markers, they are restriction enzymes. What are selectable markers? These two genes, ampicillin resistant gene and tetracycline resistant, they are selectable markers. Okay, now ampicillin resistant, tetracycline, they are antibiotic resistant genes. Yes, they provide resistivity to antibiotics. Resistance to antibiotics, so I can say they are. So this becomes your answer. Question number 8, D is your answer. Works ma'am, works fine. So question number 9, for transformation, microparticles coated with DNA to be bombarded with gene gun are made up of. For transformation, my dear children, for example, I have a plant cell and I want to get it transformed. So basically what do? Can I do micro injection? Basically I'll take a pipette and try to get things inside this particular nucleus, my dear children. I can do that if it was not a plant cell. Okay, if it was not a plant cell, if it was an animal cell, I could. Okay, so micro injection is a case of, what do you call it, vectorless method for animal cells. But here in the case of plant cell, they have an extra layer which is known as cell wall, and you cannot do micro injection. Okay, so basically what do? You use gun. And what do you call this method as? Biolistic. You call this method as biolistic method, and my dear children, not ballistic, biolistic method, and it is also known as gene gun method, where what you're going to do is you're going to take microparticles. Okay, you're going to take microparticles of gold and tungsten and basically cover it with DNA of your interest, and you're going to shoot it inside so that it directly goes to what? The nucleus. It's not like you can do it, the people who are experienced can do it. Okay, so for transformation, microparticles coated with DNA to be bombarded with gene gun are made up of silver platinum, wrong, platinum zinc, wrong, silicon platinum, wrong, gold or tungsten, correct, GT. Okay, greatest of all the time. You can go, I know that but yeah, for remembering it, you can do that. So this is for plant cells exclusively for plant cells. Okay. Chalo. Now, so what are the vectorless methods for plants and animals and things like that? Always remember, for plants it is biolistic method, for animals it is microinjection method, and for bacteria and all you have heat shock method or electroporation method and things like that. Okay, I already showed the answer. Okay, now question number 10th. Restriction endonucleases are present in mammalian cells for degradation of DNA when the cell dies? No. are used in genetic engineering for ligating two DNA molecules. I suppose we use DNA ligase for that. Are used for in vitro DNA synthesis? No. Are synthesized by bacteria as a part of their defense mechanism? Yes. Yes, my dear children. So basically in the bacteria, no, we found two enzymes. One was adding methyl group and one was basically restriction enzyme. So basically what is the use of that? I'll tell you. Listen to the story. So there's a bacteria. Bacteria's name is Guccipu, okay. So what happens, there is an organism, for example bacteriophage, okay, it put its genetic material inside, and you know what happens, its genetic material will slowly go and incorporate over here. But before that, this particular Guccipu, that is a bacteria, is going to release two enzymes. One is restriction enzyme, which does what? It is going to cut the genetic material of the other organism, but it can cut its own also. So to be safe, it also releases some other enzyme which will be adding methyl group to it, and when it is methylated, my dear children, the restriction enzyme would not be acting on themselves. Get it? So basically, restriction endonucleases are synthesized by bacteria as part of their defense mechanism, nothing else. So D becomes your answer for this particular question. Now, the most thoroughly studied of the known bacteria-plant interaction is cyanobacterial symbiosis with some aquatic ferns, gall formation on certain angiosperms by agrobacterium, nodulation of sesbania stems by nitrogen fixing bacteria, plant growth stimulation by phosphate solubilizing bacteria. I know if you're just seeing it in a question paper, you might not even recognize that this is from this particular chapter, but please understand, my dear children, this is the easiest clue that you have. Okay, whenever agrobacterium and whenever gall formation is there, just understand you're talking about the correct option. Okay, so agrobacterium tumefaciens is the name of the bacteria, yeah, and this agrobacterium tumefaciens has a very different type of plasmid. What is plasmid? Extra chromosomal DNA. That is, they have their chromosomal DNA which codes for protein. This does not code for anything much, it's just there, okay, sometime for protection, sometime for this, sometime for nothing. So basically over here, it is known as TI plasmid. What do you call this? Tumor inducing plasmid, and it has one thing that is known as tDNA, transfer DNA. What does it have? It has transfer DNA. So what exactly happens is that when this particular agrobacterium tumefaciens is going to infect a plant, it is going to pass down this plasmid inside, yeah, and basically passing down the tDNA, also transfer DNA as well. And this transfer DNA, my dear children, what does it do? It causes tumor or gall, okay, gall disease in the case of plant. So what did we do? We used genetic engineering, we removed this guy, tDNA, out of the TI plasmid. We put our gene of interest, and now we are using the same thing. So always remember, if you want to transfer something into plants and if you need a vector for that, it is agrobacteria for plants. For animals, what is a vector that we have? Retroviruses. Disarmed retroviruses, okay, you disarmed as in they're not going to do their normal function, they are disarmed, they are not going to cause any trouble in us, okay, that is what you mean by disarmed. So retroviruses are for animals and agrobacterium tumefaciens is for plants. So what is your answer for this? It is B. Now, two bacteria found to be very useful in genetic engineering experiments are Nitrobacter and Azotobacter, Rhizobium and Diplococcus, Nitrosomonas and Klebsiella, Asteraceae and Agrobacterium. What is the answer? Two bacteria found to be very useful in genetic engineering experiments are, it is Asteraceae and Agrobacterium. Asteraceae gives us a very important restriction enzyme, EcoR1, yeah, where E stands for the name of the genus which is there, Asteraceae, and CO stands for coli, yeah, and R stands for the strain, and this is the number of discovery that is there, so EcoR1. Okay, and Agrobacterium, as I told, it's a vector, and Asteraceae also acts as a vector at times. Yeah. Now match the organism with its use in biotechnology. Wait. Actually, this should be coming in the next chapter, but okay, it's okay. Chalo, match the organism. Chalo, Bacillus thuringiensis, BT, my dear children. BT, what does it do? BT makes organisms cry. BT, Bacillus thuringiensis, produces scribe gene, has scribe gene, and what does this scribe gene do? It produces protoxins. Toxins, but not toxic for themselves, okay. So what happens is that in the bacteria's body, this protoxin is protoxin as it is not active, right? It's just there. Now, when an organism has this bacteria, it's like yum, 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 yum. And when it goes inside, for example, I am the organism which ate the bacteria. Now, the bacteria is within me, not exactly within me. You have to suppose that I am an insect. Okay, it only hampers the insects, not human beings. Okay, so I am the insect and I ate it. What happened was inside my gut, inside my meat gut, it's a very alkaline pH. What pH? Alkaline pH. And what happens in alkaline pH? The protoxin becomes active toxin. When it becomes active, what happens? It cleaves around all my endotheliums, cells around there, and basically I die. The insect dies. So this is the usual case that happens. Okay, so Bacillus thuringiensis is cry proteins made by the cry genes, yeah, and they are insect specific. Now, Thermus aquaticus molecule. Thermus aquaticus is a bacteria. It stays at higher temperature, and basically Thermus aquaticus bacteria, when it wants to basically replicate its DNA, it uses its own DNA, that is DNA polymerases. But since it is highly thermostable, I am going to take this DNA polymerase that I extract from Thermus aquaticus and use it for my own purposes in PCR, polymerase chain reaction, because there also I need high temperature, and in high temperature there should be some enzyme that works. So I am going to borrow it from this guy. So Thermus aquaticus is going to give me DNA polymerase. Okay. Now, Acrobacterium tumefaciens, I already told you it is going to act like a vector. Okay, Salmonella typhimurium was the first recombinant DNA. With the help of this only I created, not me. I'm so sorry. The organisms. The first, just a second. Let me come back to life. Yeah, so first recombinant DNA was made with the help of Salmonella typhimurium and also E. coli. Okay, so what exactly happened was they took a part of the plasmid. Okay. That was from Salmonella typhimurium and inserted something else into that and saw that whether if this particular thing was put in some other organism, whether it became resistant to antibiotic or not. Okay, so that was being tested just to understand whether they can become resistant to antibiotics some point of time, or basically is there any mechanism and things like that. Let's not go into detail. Okay, but yes, it was used for the construction of the first recombinant DNA. If you have any confusions regarding that, if you want to know about that, do let me know in the comment section. I am there to tell the story. Okay, so this is how it goes. So Bacillus thuringiensis is for cry proteins. A goes with 4. I have only one option like that, and hence this becomes my answer also. So A goes with 4, B goes with 3, is it? Yes. And Agrobacterium tumefaciens goes with prime vector, and Salmonella typhimurium goes with oh my god, yeah, construction of the first RDNA. Yeah. Next, the correct order of steps in polymerase chain reaction is. What happens in polymerase chain reaction? First thing, you have the DNA, my dear children. You will denature this DNA. You are going to separate this DNA, that is known as denaturation, which would require almost up to 92 to 95 degrees Celsius. Okay, so it is kind of high head temperature out there. Yes, and after that, basically what do? The separated DNA, you need primers to attach, then only DNA polymerase can work, right? So this primary attachment is known as annealing. It is known as annealing, which will require around 50 to 55 degrees Celsius, okay, at high temperature itself. And then after that, basically after annealing, our DNA polymerases, which we got from Thermus aquaticus, Taq polymerase, which we call it Taq polymerase, will be making a new strand of DNA, and this is known as what? Elongation, which would be requiring again up to 70 to 72 degrees Celsius. Okay, so this is the usual thing. So if you see all the things that are there, it happens in high temperature. It's not known as elongation, it is known as extension. Okay, sorry for that. It is known as extension. Works. You're extending the DNA. So this all happens at high temperature, which is why you need to borrow DNA polymerases from a thermostable organism, and that was Thermus aquaticus. So steps are denaturation, annealing, and extensions. This will be your answer. Works? Now, which of the following is not correctly matched for the organism and its cell wall degrading enzyme? So, fungi, chitinase. Fungi cell wall is made up of chitin, my dear children. Yes, so we need chitinase. Correct. Bacterial cell wall is made up of peptidoglycan, and that is why you need lysozyme. You need lysozyme. You know what? Our tears have lysozyme, which is why basically they can kill all the bacteria out there. Okay, so plant cells, cellulase, because yeah, plant cell wall is made up of cellulose. Algae, what about algae? Do you need methylase? No. Algae, most of them, they also have their cell wall made up of cellulose. So I'll say this is a wrong option that we have. Okay, okay, okay. Now, during the process of isolation of DNA, chilled ethanol is added to. So, when you want to isolate DNA from all the other things, which is protein and things like that, obviously you are going to cleave with RNase, that is going to cut down all the RNA molecule, put protease to cleave down all the protein molecules. Now we are left with DNA, but you want to separate it out. So what do? You add chilled ethanol. When you add chilled ethanol, what exactly happens? Precipitate DNA, break open the cell to release DNA, facilitate action of restriction enzyme, remove proteins such as histone. Your answer is to precipitate DNA. The DNA gets precipitated and then you later take it out with the help of spooling. You might have seen the picture of this, yeah? That is known as spooling, my dear children. So your answer should be A. Cool? Now 17th question. The figure given below shows 3 steps A, B, C of polymerase chain reaction. Select the options giving correct identification together with what it represents. Okay. Okay. So this is A, which is basically showing probably denaturation. A should be denaturation, B should be what? B should be annealing, and C should be what? Elongation. That is the thing. Okay, now over here, B is denaturation. Can B be denaturation? No. Okay, and A is denaturation at a temperature. A is denaturation temperature 50 degrees Celsius. Denaturation happens above 90, not 90, above 92 actually. So this is wrong. Now C, extension. Extension in the presence of heat stable DNA polymerases. Do you think? Yes. Are you? Yes, that is correct. Now A is what? Annealing with 2 sets of primers. Do you think A is annealing with 2 sets of primers? No. So what is your answer for this particular question? It will be C. Extension in the presence of heat stable DNA polymerases. Exactly correct. Okay, works. Now, stirred tank bioreactors have been designed for what? Why do you think you need stirred tank bioreactors, my dear children? So that it can stir up and give oxygen. Okay, it can provide all the nutrients equally in all particular direction. That is why you need stirred tank bioreactors. So it is addition of preservatives to the product. This is why you need stirred tank bioreactors. No, you basically can add it through any port if you have any other bioreactor as well. So it is not the one. Purification of the product, my dear children. Is that the answer? No. Ensuring anaerobic conditions in the culture batch. Array, you need aerobic conditions, not anaerobic. Okay, there are aerobic bacteria, you need air. So this is wrong. Availability of oxygen throughout the process. Yes, so that's why you need stirred tank bioreactors, because they keep repeatedly stirring and basically repeatedly giving out all the important components out there, which is very much necessary for the organisms to grow. So your answer would be this. If you are asked stirred tank bioreactor and sparged bioreactor, you have 2 types of bioreactors in your NCERT. Which one is better for oxygen delivery? It will be sparged. It will be sparged. Please remember that as well. And with that, we complete biotechnology principles and processes, my dear children. Now we are heading into biotechnology and its applications. How far are you done? So, if you're done, if you've heard me until here, just let me know. Ma'am, done with biotechnology principles and processes, or principles and bioprocesses done. If you tell me like that, I'll be really, really happy. I'll understand that, okay, my children have reached until here. At least they heard what I said, yeah? Now we are going ahead with this particular thing with the first question that we have from biotechnology and its application. Bt cotton variety that was developed by the introduction of toxin gene of Bacillus thuringiensis is resistant to what? Insect pests, fungal diseases, plant nematodes, and insect predators. Insect predators as an organism that eat insects. So make sure you are doing the correct option. You're choosing the correct option. So what all do you have in here? So A, B, C and D. Just give me a second, think and let me know. Okay, so I hope you found out the answer. Yes. So my dear children, over here, Bt cotton actually works against, okay, the toxins in there works against insect pest. Okay, not nematode, not fungal or basically insect predators. It is just insect pests, that is dipterans, lepidopterans, coleopterans and things like that. For example, fruit flies, or cotton bollworms, corn borers, all those things. Okay, so your answer would be A. Next, which of the following is true for golden rice? So it has yellow grains because of a gene introduced from a primitive variety of rice. Okay, it is vitamin A enriched with a gene from daffodil. It is pest resistant with a gene from Bacillus thuringiensis. It is drought tolerant, developed using agrobacterium vector. What would be your answer for this? If you're talking about golden rice, you know golden rice has a story behind it. So people of Africa and all, they were not getting enough amount of vitamin A and things like that. You know, there was food scarcity and all. So the staple food that they had was rice. So it was thought, okay, I'm just telling you what I heard, okay, what I got to know. There might be some other part of the story as well. So it was thought that at least for enriching a good amount of vitamin A, there should be some mechanism, and that's how the new genetic engineered crop of golden rice was being made, where you took a gene from daffodil, and this particular gene was coding for vitamin A, and that's how the whole thing happened. So the what do you call, human beings up there had golden rice and they got enriched with vitamin A. So this is the story. This, if at all, even if this is not the story, you can remember with the help of the story. Yes, works. Next question. In RNAi, the genes are silenced using, what do you mean by RNAi? RNA interference. So what do you mean by RNA interference is that basically, you know, there is mRNA being formed out of our DNA, right? There is mRNA being formed. Just give me a second, children. Yes, so basically this mRNA is going to be coding for what proteins? Yeah, it is going to be translated. So it will be going in between the ribosomal units, and that's how translation everything works. So this is the mRNA's work usually. Okay, and it is single stranded, to be specific. Now what you are doing is, you are not letting this guy to be getting translated. How, ma'am? So basically we are going to introduce a double stranded RNA. What? Double stranded RNA we are going to introduce. And what will happen? From this double stranded RNA, one strand is going to pair up over here with this particular strand. One strand is going to pair up with this particular strand. Now do you think it's single anymore? No, it is not single, it is already mingled. Okay, so over here it becomes double stranded. Now do you think it can undergo translation? No. This is known as RNA interference. So basically in RNA interference, the genes are silenced using what? Double stranded RNA. So lots of children make mistake and put a single stranded RNA. So they think just this particular strand is coming anyways, right? But no, you are introducing double stranded, right? And from that double strand, one strand is going in. So it's basically double stranded RNA itself. It's not single stranded RNA. Please make sure you are not doing that mistake. So your answer would be A. Okay, now fourth, in Bt cotton, the Bt toxin present in the plant tissue as protoxin is converted into active toxin due to, I told you, I was the insect, it went to my midgut. In my midgut, what was the pH? What was the pH that brought the whole change? It was what? Alkaline pH, my dear children. So action of gut microorganisms wrong, presence of conversion factors in the insect gut wrong, alkaline pH of the insect gut correct. Acidic pH, no baba. And please remember, it's mid-gut. It is mid-gut region. Will you remember? I hope you do. This becomes your answer. Now fourth, RNA interference involves synthesis of cDNA and RNA using reverse transcriptase, silencing of specific mRNA due to complementary RNA, interference of RNA in synthesis of DNA, synthesis of mRNA from DNA. I already told you, synthesis of complementary DNA and RNA using reverse transcriptase, that is way too complicated. So let's exclude it. Silencing of specific mRNA. Yeah, that is what RNA interference is. You're silencing what? Specific mRNA due to what? You are giving a complementary RNA. Yeah, that is why it becomes double stranded. So this would be the correct answer. Interference of RNA, synthesis of DNA would be wrong. Synthesis of mRNA from DNA would also be wrong. Your only correct statement is B. Cool? So next, silencing of mRNA has been used in producing transgenic plants resistant to what? So silencing of mRNA was basically introduced when you know, there was tobacco mosaic plant. Okay, no. Sorry, there is tobacco plant, not tobacco mosaic plant. So there is tobacco plant, tobacco mosaic virus is there. This tobacco plant was infected by a nematode, an Ascalmenthes, which is known as Meloidigoin incognitia. Yeah, Meloidigoin incognitia. So this Meloidigoin incognitia was the one that was infecting the plants and killing the plants and things like that. So it's where the silencing of RNA was introduced. The RNA of Meloidica incognitia was silenced with the help of this method. So Meloidica incognitia is not bollworm, bollworms are insect, not viceroy, that is fungus, not bacterial blights also, it is what? Nematode. Nematodes basically are nothing but Ask helminthes. If you want the name of it, the specific name of the organism, it is Meloidica incognitia. Okay, so this becomes your answer. Now, match the following columns and select the correct option. Bt cotton goes with what? Bt cotton goes with bees. Bacillus thuringiensis, adenosine deaminase deficiency. What is that? That is something new from all the questions that we saw. So adenosine deaminase is an enzyme, okay, very important for us for immune response and things like that. Okay, so what happens is that some of us, okay, not us, basically some children, they lack this genes, the gene producing this enzyme, that is adenosine deaminase. Now, is that fatal? Yes, that is fatal. What can we do about it? Probably we can do enzyme replacement therapy, where we are just providing them the enzyme every now and then, but you need repeated transfusion. Or then you can do gene therapy. What happens in gene therapy? You are going to take lymphocytes from this organism, from this child, yeah, who do not have the enzyme, and then you are going to put the genes in there that is going to produce the enzyme, and basically you are going to put this lymphocyte back into the baby, the child. Okay, what happens? Until and unless the lymphocytes are there, the baby's body or the child's body will produce what? Adenosine deaminase. But once all the cells are right, what do? Again, you need transfusion. So it is not what do you call a completely curative technique that we have. What is completely curative technique then? There is the next question. So it also is a part of gene therapy, where what you are going to do is that if a person has it, you are going to take those genes from the bone marrow and you are going to put it in the embryonic stage itself. You are going to provide the child in the embryonic stage itself this particular gene. In that case, my dear, embryonic cells, they divide very fast and they incorporate everything. Okay, they are totipotent, they will be dividing and giving this genes to all the other cells which are going to be formed out of it. At that stage, if you treat, it's all curative. Otherwise, it's not curative. Please understand that. Okay, so adenosine deaminase deficiency, you're gonna try to cure it by gene therapy. Okay, RNA interference is a cellular defense. Yeah, I forgot to tell about that. So this is so RNA interference, now we use it for a general sense also. We put double-stranded RNA and then we wait for the organism to come, and that's how it usually works. But some organisms have it in their own body as a defense mechanism, and those organisms are nothing but eukaryotes. Prokaryotes do not have it, please remember that. It's just the eukaryotes which have RNA interference as what? Cellular defense mechanism. Okay, now what do you mean by PCR? PCR stands for polymerase chain reaction, and PCR is something that can be used for finding even the trace amounts of pathogens in the body. Very little amount of pathogens there, we do PCR, because in PCR what do? We amplify it, we increase the number, right? So that's why we get to know, okay, I can test and I can randomly take a test of all these things that is up there, and I'll identify that, oh, this particular person has this particular disease. If it is found in trace amounts, my dear, there should be some method to amplify, right? Otherwise we would not be able to do correct analysis. So it helps in correct analysis, and that's why we say PCR is there for detection of HIV infection as well. Okay, works. So Bt cotton goes with 4. So I just have one option that goes with A, goes with 4. Okay, B goes with adenosine deaminase deficiency, goes with 1, that is gene therapy. RNA interference basically goes with cellular defense, and PCR will be going with detection of HIV infection. Okay, works. Next, which of the following statement is not correct? In man, insulin is synthesized as a pro-insulin. Do you think in man insulin is synthesized as pro-insulin? Yes, that is correct. It is not actually the mature insulin that it produces directly. It is pro-insulin. And what is the difference between pro-insulin and natural insulin? So in pro-insulin, there is A strand, there is A peptide, there is B peptide, and there is something joining in between, that is C peptide, my dear children. So what this is pro-insulin. Now when you are removing C peptide, when you just have A and B and they are joined with the help of disulfide bridges and things like that, it becomes what? The mature form of insulin. Okay, so in man, insulin is synthesized as pro-insulin. Correct, ma'am. The pro-insulin has an extra peptide called C-peptide. Correct, ma'am. The functional insulin has A and B chains linked together by hydrogen bonds. It is disulfide bond. So this is wrong statement. Genetically engineered insulin is produced in E. coli. Was it? Yes. Eli Lilly company. Okay, there is a company that is Eli Lilly. It's still there. It's a very famous company. Now they were the ones who put down insulin. Okay, and this genetically modified insulin or genetically engineered insulin was known as Humulin. It is known as Humulin, my dear children. So what did we do? We took Escherichia coli, 2 of them. Okay, suppose in one you put A peptide, in other you put B peptide. Both of them produced it, A and B, and then you joined it in the laboratory condition. Why didn't you put the exact gene into it? Because if we put the exact whole gene, just like as it is there in the case of human beings, it will also produce C peptide. So in our body there is an option to cut this C peptide, but do they have it? No. So that is why it was introduced separately. Okay, so and the thing that was produced was known as Humulin. So your answer for this question is C. You are asked what is not correct. This is not correct because it is not hydrogen bonds, but it is disulfide bonds. The first human hormone produced by recombinant DNA technology is what? Again, insulin. Nothing else but insulin. Now next, maximum number of existing transgenic animals is of fishes, cow. Transgenic animals, what do you mean by transgenic animals? For example, I am bringing out changes in their genetic makeup, my dear children, by my own will, by genetic engineering. These animals are known as transgenic animals. I can use it for studies, I can use it for production of certain things that I want. Okay, all these things. So what is the maximum number? Which one was the most used one? Is it fish, is it cow, is it pig, or is it mice? It is mice. Talking about pig, so initially humans were given pig insulin to the pig, but some human beings were becoming very allergic to it, which is why we thought that we should be doing something about that, and that's how Eli Lilly came up with this particular idea. So it is mice that is being used the most. So read the following 4 statements A to D. The first transgenic buffalo, Rosie, produced milk which was having human alpha-lactalbumin, enriched. Ma'am, Rosie name have read, and alpha-lactalbumin also I have read. So this should be the correct answer. No, ma'am, this is a correct statement. So you have to find the statement which has mistakes in it. And I see that a big mistake is there. It was not buffalo, my dear children. Okay, we are buffalo. To think like that, it was actually a cow. The cow's name was Rosie. You think how bad the cow might have felt. Okay, after putting their name Rosie, now you have changed the cow to buffalo. My God. So A is the thing that is wrong. Restriction enzymes are used in isolation of DNA from other macromolecules. When did people start using restriction enzyme for isolation of DNA? I thought it was just cutting the DNA strands and for using it for genetic engineering, things like that. So again, this is also wrong. This is wrong, this is wrong. Downstream processing is one of the steps of rDNA technology. Downstream processing means you are purifying it, you are separating it. These steps are known as downstream processing. Is it a part of recombinant DNA technology? Yes, ma'am. Correct. D. Disarmed pathogen vectors are also used in transfer of rDNA into the host. Disarmed pathogen vectors, do we use disarmed pathogen vectors, as in for example retrovirus is there, it usually causes trouble in our body, but now we are disarming, that means they are not going to cause any trouble in our body, they are just there for transferring things up. Okay, so disarmed pathogen vectors are also used in transfer of rDNA into the host. That is correct. That is correct. So what is not correct? A and B would not be correct. A and B are the ones that has mistakes in it. Okay, please remember that. Yes. Now question number 12. A new variety of rice was patented by foreign company, though such varieties have been present in India for a long time. This is related to a new variety of rice, my dear children. It was patented, okay, by a foreign company. So what do you call this? But it was already there. What do you call this as? No, it's not what do you call this as. What was this about? So it was already there, but it was patented by some foreign country and company actually. It was basmati rice. It was basmati rice, not CO667, not Sharbati Sonora, not Lerma Rojo. It is basmati rice. Simplest answer that we have over here. In India, the organization responsible for assessing the safety of introducing genetically modified organism for public use is what? Indian Council of Medical Research, ICMR, Council for Scientific and Industrial Research, CSIR, okay, Research Committee on Genetic Manipulation, RCGM, or Genetic Engineering Appraisal Committee. It is genetic engineering appraisal committee which give the rights that okay, I can use this particular organism for doing genetic engineering, and also giving rights that okay, whatever I have made, I can use it for public, I can make it public also. You know, all the rights have been governed by this particular association, that is genetic engineering appraisal committee. You might see an approval committee also, both of them are equally correct. The name has been changed to approval committee now, but yes, this was the initial one. This is there in your NCERT, so please remember it. Now, the use of bioresources by multinational companies and organizations without authorization from the concerned country and its people is called use of bioresources, my dear children, by multinational companies. You are using by multinational companies and you are not giving proper authorization and things. Okay, for example, I am the multinational company. I am from belong to different country. Okay, now I have been using India's rice and I am taking patency to it, that only I can sell this, no one else can sell this. What do you call this? This is totally bad, right? This is their product, I am using it. I am the bad person. So what is it? What is this process known as? This is known as biopiracy. Okay, it is known as biopiracy because if I probably gave authorization or probably I asked their concern and everything and then I did something, it would have made sense, but over here I am not taking anything, I am doing everything without authorization, so this will be known as biopiracy. 14th one, your answer is B. And with that, we complete biotechnology and its applications as well. If there is something that you have doubt, if you have any doubt in anything, please let me know in the comment section. If you want concept clarity in some more things, you can ask me. Okay, again, I can probably arrange a video for that, okay, if you ask me. So make sure you're putting down all these things and do let me know if you have reached this particular point. How do you say so? Ma'am, I have completed 2 chapters. Yes, say that. That's all. Now, human health and diseases, the first question that we have over here, the infectious stage of Plasmodium that enters the human body is. What is the infectious stage of Plasmodium that enters the human body? In which stage does it enter? Sporozoite stage, my dear children. It is sporozoite, not trophozoite, not female gametocytes, not male gametocyte. It is just sporozoite. And where is it exactly? Just imagine a mosquito like this. Okay. So basically, in the salivary glands of the mosquito, it has sporozoites, and when it comes and bites, always think like this. Okay, so some people think that they will this mosquito bit a person with HIV virus and now it comes and bites me, I will also get HIV virus. I should not be going near that person. Is it like that, my dear children? Mosquito is not a fool to give back the blood that it took. Always remember that. Okay, so it's there in its body. It's not going to give back the blood. Basically, what it does, it is releasing its saliva because it has certain anti-clotting factors, which is why it can drink the blood peacefully. So in the salivary glands, you have sporozoites, and these sporozoites are going to travel down with the help of saliva now into the blood of the human body. Okay, from the saliva goes to the body, from the body in the blood circulates, goes to the liver, then goes to the RBC, cause ruckus out there. Okay, so basically it's sporozoite stage where it enters the human body. And next, which of the following sets of diseases is caused by bacteria? Cholera and tetanus, typhoid and smallpox, tetanus and mumps, herpes and influenza. What is your answer for this question? So ma'am, in our NCERT, about cholera and tetanus and all, they have not mentioned. How can we tell the answer for this? Ma'am, you have to know something outside your NCERT also. Yes, so typhoid is mentioned, diphtheria, dysentery has been mentioned, and then you have pneumonia that has been mentioned. Okay, so cholera and tetanus is also something that is caused by bacteria, please remember it. And smallpox is caused by a virus, mumps is caused by a virus, herpes is caused by virus, yeah, and influenza is also caused by virus. Or influenza, if you're talking about, no. You're not talking about that influenza, yeah. Now, so cholera, Vibrio cholerae, okay, it is a comma shaped thing, that is bacteria. Tetanus, Clostridium tetani, that is a bacterium. Typhoid, Salmonella typhi, that is a bacterium. Smallpox, variola virus, that is virus. Mumps virus, okay. Herpes simplex virus, that is herpes, that is a virus. Influenza virus, that is virus, okay. So it's cholera and tetanus that becomes your answer for this particular question. Now, motile zygote of Plasmodium occurs in. Motile zygote and Plasmodium. So I'll tell you one scenario. So in the human body, I told you it's the liver that gets affected first. So okay, the liver. The cells of the liver, they break open and things like that, and basically now they will be moving to RBCs. In the RBC, my dear children, they are going to the Plasmodium which is there. It will be in some stage, yeah, and it is going to basically feed on hemoglobin and break it down into heme and globin. Heme and what? Globin. This heme is kind of toxic for them, so they convert it into hemozoin. Okay, this is one scenario that is happening up there, but it is all happening in the RBC. Apart from that, they are also transforming into next stage, which is known as gametocyte stage. Now what happens? Everything is happening inside the RBC. The RBC will burst open one day and gametocytes will be released, and also hemozoin will be released. This hemozoin is kind of toxic for us. Okay, they kind of made the toxic version of something into something that is toxic for them, but it is actually toxic for us. Hemozoin is toxic for us, which is why we get fevers and chills, right? Now gametocytes are stages which will later form gametes. But do they form gametes in the human body? That is the question. Do they form gametes in the human body? The answer is no. Where are these gametocytes now? In the blood. What will happen? Another mosquito comes, takes in this gametocyte. The blood will be going into the gut of the mosquito. In the gut of the mosquito. Now what happens? The gametocytes will form gametes. The gametes will be fusing. It will be forming something that is known as ookinete stage. Okay, ookinete. And in this stage, it is going to penetrate through the wall and basically move upwards and form sporozoite. So where do you think motile zygote of Plasmodium occurs? In the gut of the female Anopheles mosquito, and that is known as ookinete, Always remember that, okay? So it is in the gut of the female Anopheles mosquito. It's not in the salivary gland of the Anopheles mosquito. In the salivary gland, what do you get? Sporozoite stage. Just after ookinete stage, you get sporozoite stage. Human RBCs, no. Human liver, no. So sexual reproduction, is it happening in the human body? The answer is no, it is happening in the gut of the mosquito. Now question number 4. A person suffering from a disease caused by Plasmodium experiences recurring chill and fever at the time when? Now if you are not answering this question, I cannot do anything. So A, the sporozoites released from RBCs are being rapidly killed and broken down inside spleen. That is not why we get fevers and chills. The trophozoites reach maximum growth and give out certain toxins. No, the parasite, after its rapid multiplication inside RBCs, ruptures them, releasing the stage to fresh RBCs. Correct. Parasite, after rapid multiplication inside the RBCs, rupture them, releasing the stage to what? Fresh RBCs. And when they do so, they are also giving out what? Hemozoin, and that hemozoin gives fevers and chills. Okay, the macrogametes and megagametes are being destroyed by the WBCs. Wrong. So your answer becomes what? C. Next, match the disease in column 1 with the appropriate items, pathogen prevention and treatment in column 2. Okay, amoebiasis, diphtheria, cholera and syphilis. Okay, amoebiasis, my dear children, is caused when you have contaminated food. Okay, it is occurring through the fecal-oral route. Okay, so all this contaminated food which is there, suppose a fly comes and sits on that, and then comes and sits on basically, what do you call it, on the food, my dear children. You can get the disease and things like that, or if you do not wash your hands and things like that. Okay, so amoebiasis, please use only sterilized food and water. Diphtheria, you have DPT vaccine. Okay, cholera, my dear children, cholera is when you get loose motion, ultimate level of loose motion, my dear children, very bad. So you would be dehydrated so very much, so you need oral rehydration therapy for that. Okay, now syphilis is caused by Treponema pallidum. Okay, syphilis is caused by Treponema pallidum. And with that, we end this. So amoebiasis with 2. A goes with 2. I have so many options like that. Okay. Let's see D. D goes with 1. D goes with 1. How many options like that? 1. And hence I got the answer. This is how we do fast measurement. Okay, fast answering. So over here, A goes with 2. Okay. B goes with 3. And C goes with 4. And D goes with 1. And that becomes your answer. Okay. Which one of the following is not correctly matched? Glossina palpalis, sleeping sickness, Culex pipiens, filariasis, Aedes aegypti, dengue fever, Anopheles crucifacius, leishmaniasis. So yes, Glossina is a thing that we have, insect that we have, and this particular one carries what do you call, trypanosoma, that causes the sleeping sickness, my dear children. Culex is the one that causes filariasis. It carries Vicariaria bancrofti, Vicariaria malai. Aedes aegypti is something that causes dengue fever. Okay. Now Anopheles is not something that causes leishmaniasis. Anopheles causes malaria. Okay. Anopheles basically causes what? Malaria. It carries Plasmodium. Now, yeah. Now, leishmaniasis is caused by leishmania, and it is carried with the help of sandfly. It is carried with the help of sand fly. Fine. So this would be wrong. So here this option is wrong. Next, identify the wrong statement with reference to immunity. When exposed to antigen, living or dead, antibodies are produced in the host body. It is called active immunity. When exposed to antigen, okay, antibodies are produced in the host body. It is called active immunity. Yes, your body is actively participating and it is reacting to it, which is why you have antibodies. This is known as active immunity. Yes, correct. When ready-made antibodies are directly given, it is called passive immunity. Yes, you are not allowing your body to produce, you are basically directly giving it, so it is known as passive immunity. Correct. What do you have to find out? You have to find the wrong statement, right? Now, active immunity is quick and gives full response. Now, whenever you want your body to work on something, my dear children, it will be a slow response. It cannot be quick. Okay, some antigen came, my body is seeing it for the first time. Obviously, it will take some time to produce antibodies. So, active immunity is actually slow. If you want it fast, you do passive immunity. For example, if some person is getting a snake bite, you cannot wait for your body to produce antibodies against it. Rather than you will just go there and give some antivenom injection that contains preformed antibodies. That is passive immunization, my dear children. You get it? So that would be fast, but active immunity is not going to be fast. Please remember that this is a wrong statement. Now, fetus receives some antibodies from mother. It is an example for passive immunity. Yes, you are getting ready-made antibodies. It is an example of passive immunity. What are the antibodies if you are asked? From the placenta, you are going to receive antibody G. From the mother's colostrum milk, which is there, my dear children, colostrum, you are going to get A. So always remember A for Amma and A for the antibody that is being given from the mother's milk. Okay, so first antibody to go is through placenta, and that is IgG. IgG. How many antibodies do we have? GAMED, gamed. Okay, so G over here is the first one that passes through the placenta, and A passes through the colostrum. Okay, so this is also correct. Your only incorrect option is C. Now, which of the following immune responses is responsible for rejection of kidney graft? As when you're talking about whenever you are getting a graft, what type of immunity will be the one that will be initiating a rejection? Cell-mediated immune response, autoimmune response, humoral immune response, inflammatory immune response. What is the answer? It will be cell-mediated immune response, where you are basically focusing on T lymphocytes. So T lymphocyte plays a major role over here, and they are going to help with cell-mediated immune response. It's not autoimmune response, it is not humoral. In humoral, you have B lymphocyte that plays a major role. Okay, so it is cell-mediated immune response that helps in kidney graft and all kind of graft rejection. It is by T lymphocytes. Now, MALT, mucosa associated lymphoid tissue, constitutes what percent, about percent of this percent of lymphoid tissue in the human body? First of all, what do you mean by lymphoid tissue? Lymphoid tissue there are two types of lymphoid tissue, primary and secondary. Okay, or lymphoid organs, primary lymphoid organ, secondary lymphoid organ. Primary are bone marrow and thymus. So you might know bone marrow is a place where all the cells are being formed because they have the stem cells. So all the red blood cells, white blood cells, platelets, all of them are being formed. In white blood cells, you include the B lymphocytes and T lymphocytes, so they are also formed. So they form, they mature. Do all mature there itself? B lymphocytes mature in the bone marrow. Okay. T lymphocytes goes to the thymus for maturing, which is why thymus is also known as primary lymphoid organs, because formation, maturation and antigen sensitization occurs in primary lymphoid organs. That means they become sensitized to antigen. That okay, if this particular antigen comes, I have to attack. This is known as antigen sensitization. So this things happens in the case of primary lymphoid organs. From there, after the training, they are going to actually do the attack. Okay, they are going to actually do the work. Where in the secondary lymphoid organs, what are those? Spleen, lymph nodes, my dear children, and MALT, mucosa associated lymphoid tissue. Mucosa is the innermost layer that we have in the gastrointestinal tract, and everything that is associated with that has the lymphocytes are known as mucosa associated lymphoid tissue. That is your tonsils over here. Inside you have tonsils, in the intestine you have Peyer's patch and all these things. Okay, so they are going to fight back against this because they have the the lymphocytes over there, and there they are going to be the effector cells. Okay, so over here formation, maturation and basically antigen sensitization. Over there they are going to become the effector cells. They are going to fight back against all this. So in MALT, which includes the tonsils and Peyer's patch, it includes 50%. MALT constitutes about dash percent of the lymphoid tissue. It is 50% of the lymphoid tissue. Do not forget that. Now, Which of the following is correct regarding AIDS causative agent HIV? So HIV is enveloped virus containing one molecule of single-stranded RNA and one molecule of reverse transcriptase. Answer wrong. Okay, why? Because it has, okay, single-stranded is there, but there are two of them. They are not mingled, okay, they are separate, but there are two of them, my dear children. This is wrong. And they do not have just one molecule of reverse transcriptase, they have two reverse transcriptase molecules, and apart from that they have two more enzymes. One is protease, another one is integrase. Integrase is for integrating into the viral genome, sorry, human genome, and protease is basically for cleaving around and doing all the other functions. Yeah, so HIV is enveloped virus that contains two identical molecules of single-stranded RNA, yes, and two molecules of reverse transcriptase, absolutely correct. HIV is enveloped, wrong. They have a capsid. HIV does not escape but attacks. Wait. Which of the following is correct regarding HIV does not escape but attacks the acquired immune response? It does not attack, actually it evades, but I'll say both of them are correct. Okay, both B and D are almost correct. If you want to choose the better option, I'll say this is the option. Okay, HIV does not escape but attacks. Instead of attacks, we can say it evades. Yeah, it evades, it gets inside and basically damages the T lymphocytes and macrophages and all those things. But yeah, both of them would be correct. Okay, B and D would be equally correct. I'll say this one has more preference, so please go for it. Okay, it does not escape, actually it does not escape. So yeah. Now, which of the following techniques is safest for detection of cancers? Magnetic resonance imaging, radiography, computer tomography, and histopathological studies. Histopathological studies, you have to cut down some portion of the part and then you have to study it. Okay, radiography, you're going to use ionizing radiation. Computer tomography also, you're going to use X-rays, that is ionizing radiation. Magnetic resonance imaging, you're just going to use magnetic field, and also non-ionizing radiation, which is comparatively safer. My dear children. So your answer would be A. Okay, next. Which is a particular type of drug that is obtained from the plant whose one flowering branch is shown over here? What is this? This is Datura. This is Datura, and Datura, my dear children, Datura, Atropa belladonna, cocaine, all of them, they are hallucinogen. They make you hallucinate. Okay, you will feel that elephants are flying. Okay, you will feel that you are flying, or probably you are flying, and all this hallucination can happen, which might sound fun, my dear children, but the thing is, it is not actually really fun. Why? Suppose you are crossing a road, you had some hallucinogen, you are not able to visualize what is actually happening, you cross the road, you feel you are flying. You get it? So these are things which is why all these things are banned. It's really bad. It goes really bad. That's why. So which is a particular type of drug that is obtained from the plant whose one flowering branch is shown over here. It is hallucinogen, and this plant is Datura. It is not depressant. There is one depressant given in your NCERT, and that is heroin. There is one painkiller given in your NCERT, that is morphine. It's a painkiller and a sedative. Stimulant? Nope. Okay, it's not a stimulant as well. Next. Identify the molecules A and B. This is the last question, okay? Molecules A and B shown below, and select the right option giving their source and use. So over here, what is this, my dear children? Ma'am, it's not there in NCERT, I know. What about this? This is there in NCERT, right? Can you tell what is this? B is what? It is cannabinoid, like ganja, marijuana and all this thing. Where do you get cannabinoids from? Cannabis sativa. What do? They affect your they affect your cardiovascular system. Okay, please remember, they are going to affect your cardiovascular system. Cannabinoids are going to affect your cardiovascular system. Remember that. You have all these examples as well. Okay. Now, what about opioids? Opioids basically are going to affect your CNS, central nervous system, and the gastrointestinal tract. What about coca alkaloids, cocaine and all this thing? Again, CNS, and it is going to interfere between the dopamine transfer and things like that. So this one is a cannabinoid. Is what? A cannabinoid. One is given in your NCERT. So two pictures are given. One is of this, one is of opioid, which looks like an O. Morphine structure is given, that looks like an O, so that is easily identifiable. And this is what? This is your, what do you call it? Cannabinoid. What is this? This is cocaine. This is cocaine, my dear children. Okay, so you can remember crow if you want. This looks like a bird, right? And cocaine, you get it from Erythroxylum coca. And what does it do, my dear children? It accelerates the transport of dopamine. So there are dopamine receptors, is basically closest of all of them, such that the dopamine do not have a place to go. It will keep on moving ahead. Okay, so this is the thing about dopamine, and this is what it does. And what happens when someone have it? Basically, they have a sense of euphoria, because dopamine is a happy hormone. It makes you really happy. So dopamine is totally transmitted every now and then, which is why the person becomes happy, they hallucinate, and all these things happen, which is not really good, which is why cocaine is actually banned. So it looks like this, and that is cocaine from Erythroxylum coca and all this thing. B option is not given over here. It is, okay. So B cannabinoid is given, but it is not from Atropa belladonna. You get it from cannabis sativa. So that would be wrong, and does not produce hallucination usually. So that is also wrong. Okay. So that's all, my dear children. There are lots of other things as well. What is important? For example, tell me the name of the, okay, if you know, can you please tell me the name of the drug that increases the blood pressure? Okay, that increases the blood pressure. It is found in cigarettes. Now you can try to, cigarette is something that has the component which would be having that component that will be causing increase in blood pressure. So yeah, that is there. Apart from that, yeah, so some sportsmen, they abuse cannabinoids. They take cannabinoids, okay, for good performance and things like that. Apart from that, they use anabolic steroids, and because of this anabolic steroids, they have different problems in men and women. For example, in women, there is enlargement of clitoris, there is irregular menstrual cycle, there is mood swings, there is depression and all this acne and all this thing. In the case of male, there is enlargement of prostate gland, the reduced size of testicles and all these things. Okay, again mood swings and these things are there. So these are few other things that is there. Now what comes under innate immunity? Innate immunity is what non-specific time of immunity is there with us from childhood when we are born. Okay, that is innate immunity, my dear children. So all this small information are really important. Cytokine barriers, cellular barriers, physiological barriers, physical barriers. Okay, examples of that. What is the immune modifier in the case of cancer? It is alpha interferons. Alpha interferons are given to the cancer patients so that the immune system becomes active. Okay, and there is another interferons that is released from viruses, virus infected cells. Okay, so they release, virus infected cells release interferons and tell the other cells that you have to be safe, and that is cytokine barrier that comes under innate immunity. So all this little piece of information is also important. Please make sure you're going through all this thing. Okay, and what antibody structure, two heavy chains, two light chain, there is variable region and there is constant region. In the variable region, you will be having area for an antigen attachment as well, antigen binding site as well. Yeah, and the name of all the diseases, organisms that causes it, please have it in your head. It will be really important. Yeah, so my dear children, I hope this session was helpful for you. Do let me know the name of the drug in the comment section below, and wishing you all the best. Please practice it thoroughly as much as you can. Give priority to all the questions. I just wanted to explain at least few concepts so that if at all these questions came, you can easily answer all of this. Yeah, so wishing you all the best. Take care, and if you like the video, definitely do let us know in the comment section. Also like and share and subscribe the channel. Okay, if you have not subscribed it yet, and please share it with your friends. Yeah, so bye bye, take care, lots of love.